Investigating the role of SGLT2 in altered metabolism of prostate cancer. So, the prostate is a small walnut sized gland located in the pelvis beneath the urinary bladder and in front of the rectum of the male. And its major role is to add fluid to semen during ejaculation to allow sperm to survive outside the male body. So, the prostate cancer among US men is the number one diagnosed non-cutaneous cancer and the third leading cause of cancer death. American Cancer Society projects that there will be around 161,000 new cases of prostate cancer and around 27,000 deaths in 2017. Additionally, 1 in 7 men will be diagnosed with prostate cancer during his lifetime. Although the etiology is not well understood, some of the risk factors for prostate cancer involve genes, such as changes in genes that regulate androgen action. And this is important because androgens not only stimulate the growth and development of normal prostate cells, but also prostate cancer. Men who are more than 50 years of age are at an increased risk for prostate cancer, and around 6 in 10 cases, prostate cancer occurs in men that are more than 65 years of age. Additionally, men who eat a lot of red meat and high fat dairy products and less fruits and vegetables are shown to be at an increased risk for prostate cancer. African American men and men in the Northern Americas, Europe, and Australia are at an increased risk for prostate cancer. So, the treatment for prostate cancer, for localized prostate cancer, involves watchful waiting and active surveillance, for which the patients are closely monitored and they're given treatment only when there is changes in symptoms. There's also prostatectomy that involves complete removal of the prostate, radiation therapy to use radiation to kill the cancer cells as well as the cells surrounding it, and cryotherapy that uses supercooled gases to freeze the prostate tumor as well as the cells surrounding it. When the prostate cancer metastasizes to a different organ, then it is called advanced prostate cancer, for which the first line therapy involves the hormone sensitive therapy or the hormone therapy, for which surgical or chemical castration is done. So the surgical castration involves complete removal of the testes so that the testosterone levels go down, and chemical castration involves using drugs such as luteinizing hormone releasing hormone antagonists or LHRH antagonists that block the testosterone production from the testes. And even though these treatment approaches are very effective, um, there is emergence of castration resistant prostate cancer for which these treatment approaches fail. And in those cases, anti-androgens can be used, that involves drugs that can inhibit the enzyme that promote androgen synthesis as well as androgen receptor blockers. And the major region for castrate resistant prostate cancer is thought uh, to be the prostate cancer cells being able to produce their own androgens, or that they upregulate the androgen receptors. Sometimes even these treatment approaches fail, leading to hormone refractory prostate cancer, for which the only treatment available are chemotherapy and vaccine treatment. So there is clearly a need for novel therapies with novel mechanisms of action to target specifically the advanced prostate cancer. And so the effort to develop novel anti-cancer therapies have targeted on metabolism of cancer cells. For instance, in the normal differentiated cells, they utilize so they convert glucose to pyruvate in presence of oxygen, and this pyruvate is further oxidized in the Krebs cycle to produce energy. And in absence of oxygen, the glucose is converted to lactate, a process known as anaerobic glycolysis. However, in highly proliferative cells and in tumor cells, even in the presence or absence of oxygen, the glucose is primarily converted to lactate, a process known in presence of oxygen known as aerobic glycolysis. And in order to meet the high demand for the highly proliferating cells, the cancer cells exhibit a high glucose consumption and glycolysis rate, a process known as the Warburg effect. So, targeting the metabolism of cancer cells has centered on glucose transporters, um, especially the GLUT1. That high GLUT1 expression has been seen in various cancers, including the prostate cancer. And the PET scan based on the 2-deoxy 2-fluoro-D-glucose or 2-FDG uptake has been utilized for diagnosis of various cancers. However, this method cannot reliably detect prostate cancer, and so this group from UCLA suspected that there might be another glucose transporter involved. And here I'm showing the hematoxylin and eosin staining of tissue sections from human prostate adenocarcinoma samples, where you can see tightly packed microacini with irregular contours, one layer of epithelial cells, irregular epithelial cells compared to two layers of epithelial cells in the normal prostate tissue, and as well as high nucleus to cytoplasm ratio, all characteristics of cancerous tissues. And what this group showed was that SGLT2 is predominantly expressed in the malignant epithelial cells. And so SGLT2 belongs to the sodium glucose transporter family, and they are predominantly expressed in the first segment of the proximal tubule of the kidneys in the apical side facing the lumen. And the SGLT2s, they utilize the electrochemical gradient of sodium generated by the sodium potassium ATPase pump in the basolateral membrane to shuttle the glucose against their concentration gradient by active transport. So there are also around 6 major members of the SGLT family, with new members emerging, and all of these SGLTs utilize various or they transport various substrates such as glucose, galactose, mannose, and inositol, and they are distributed throughout different uh, organs in the body. The most widely studied are SGLT1 and SGLT2, and as mentioned before, SGLT2s are predominantly present in the first segment of the proximal tubule of the kidney, with very limited expression in brain, liver, thyroid, muscle, and heart. SGLT1 is predominantly expressed in the small intestine, as well as the second and the third segment of the proximal tubule of kidney. And something to note here is that SGLT1 is also expressed in the normal prostate tissue. And here I'm showing the autoradiographic images showing Me4-FDG uptake. And so Me4-FDG is a form of glucose that is specific to SGLTs but not glucose transporters. And what you can see here is that Me4-FDG is transported into the human prostate adenocarcinoma tissue in vitro. And this Me4-FDG uptake is blocked by this SGLT2 specific inhibitor called dapagliflozin, as shown by the reduction in these radiographic image. So what this suggests is that SGLT2 contributes to glucose uptake in human prostate adenocarcinoma in vitro. And here I'm showing the PET-CT images of the PC3 xenograft mouse model of prostate cancer. And PC3 is the prostate cancer or the adult human prostate adenocarcinoma tissue cell line. And here what you can see is that ME4-FDG, which is a substrate for SGLT, is can be used to detect prostate cancers in vivo, as shown by this as shown by this region of glucose uptake in the xenograft mouse model. And this glucose uptake is blocked by this SGLT specific inhibitor dapagliflozin, suggesting that SGLT2 contributes to glucose uptake in prostate adenocarcinoma, also in vivo. The specificity of SGLT2 inhibitor here is also shown by this ME4-FDG um, region in this urinary bladder, suggesting that the SGLT2 was not reabsorbed by the kidneys. So the study rationale for this proposal is that SGLT2 is functionally expressed in human prostate adenocarcinoma. Unlike the glucose transporters that are expressed abundantly throughout different tissues in the body, SGLT2 is predominantly expressed in the kidney with limited expression in some other tissues. Additionally, SGLT2 was shown to contribute to glucose uptake, tumor growth, and survival in pancreatic cancer xenograft mouse model. However, whether SGLT2 can play a similar role in prostate cancer has not been studied and will be the focus of the study. So my hypothesis 1 is that SGLT2 contributes to altered metabolism of prostate cancer cells, and hypothesis 2 is that FDA-approved SGLT2 inhibitor drugs can provide beneficial effects in prostate cancer treatment. My aim 1 is to investigate the role of SGLT2 in altered metabolism of prostate cancer cells, and my aim 2 is to investigate the potential of SGLT2 inhibitors as a therapeutic for prostate cancer. And here are the list of SGLT2 inhibitors approved by FDA and in use as a third-line therapy for the treatment of type 2 diabetes. And their structures and IC50 values for inhibition of SGLT2 are given here. What I want to point out here is that dapagliflozin and empagliflozin show a very high selectivity for SGLT2 against all the other SGLTs and glucose transporters. However, canagliflozin has been shown to partially inhibit GLUT1 and SGLT1 as well. So my aim 1.1 is to first evaluate the SGLT2 mRNA and protein expression levels in normal and cancerous prostate cell lines. So SGLT2 mRNA and protein have been reported in this human prostate adenocarcinoma derived cell line PC3, and SGLT2 mRNA is absent in the normal human prostate tissue, and nothing has been reported about the SGLT2 protein. Additionally, whether SGLT2s are expressed in other prostate cancer cell lines as well as normal prostate cells have not been studied, and thus I will evaluate the SGLT2 mRNA and protein expression in normal prostate cell lines such as RWPE1 and PNT2, and prostate cancer cell lines such as DU145 and RWPE2. My aim 1.2 is to investigate the role of SGLT2 in altered metabolism of prostate cancer cells. And as mentioned before, dapagliflozin, which is the SGLT2 inhibitor, was shown to exhibit reduced glucose uptake in human prostate adenocarcinoma samples and PC3-induced xenograft tumor in mice. Additionally, in another study, canagliflozin, which also partially inhibits GLUT1 and SGLT1, has been shown to reduce glucose uptake, clonogenic survival, and cell proliferation in PC3 cells. However, since canagliflozin also partially inhibits GLUT1 and SGLT1, I will use the more specific inhibitors dapagliflozin and empagliflozin to investigate the role of SGLT2 in altered metabolism of prostate cancer cells. So the PC3 cells will be treated with 250 nanomolar of dapagliflozin and 100 nanomolar of empagliflozin. The concentrations have been derived from, so these concentration has been shown to reduce glucose uptake either in the human prostate adenocarcinoma tissue themselves or in kidney cells. And I will use 100 nanomolar gemcitabine, which is an anti-cancer chemotherapy drug, as a positive control, and there will also be a vehicle control. I am interested in metabolic parameters such as glucose consumption, glycolytic flux, cell proliferation, ATP content, cell death, chronogenic potential, citrate oxidation, and lipogenesis. So PC3 cells have been shown to exhibit high glucose consumption and glycolysis compared to normal prostate cell line RWPE1, in line with the Warburg effect. And I hypothesize that since SGLT2 are glucose transporters, their inhibition will lead to reduced glucose consumption and glycolysis in PC3 cells. To study glucose consumption, I will utilize a glucose assay kit, which is a luminescence assay, and the glucose consumption will be calculated by glucose in the sample versus the glucose in the cell-free media. The glycolytic flux will be measured by monitoring the tritium incorporation to water from the tritium labeled glucose in the second last step of glycolysis, and will be normalized to radioactivity of tritium labeled glucose. As mentioned before, SGLT2 is involved in glucose transport into PC3 cells, and since glucose provides the ATP and carbon for macromolecule biosynthesis, I hypothesize that SGLT2 inhibition will lead to reduced cell proliferation and ATP content and increased cell death in PC3 cells. Additionally, I will also evaluate the chronogenic potential of PC3 cells upon SGLT2 inhibition, which is a characteristic of cancerous cells. To calculate the cell proliferation, I will count the viable cells using trypan blue exclusion, and I will alternatively also stain the cells for Ki67, which is a marker for proliferation. For ATP content, I will utilize the ATP bioluminescence assay, which utilizes the conversion of luciferin to luminescent signal by luciferase, and this reaction requires ATP, so the increase in luminescence is directly proportional to the ATP content, and the ATP content will be normalized to cell density. Cell death will be measured using annexin V and propydium iodide staining. Annexin V basically binds to the phosphatidylserine on the outer surface of the cell membrane in the apoptotic cells versus the inner side in the healthy cells. And propydium iodide is a cell impermeant dye and will allow us to distinguish between the early apoptotic cells, which are membrane intact, and the late apoptotic cells, which are membrane ruptured, and also necrotic cells, which are membrane ruptured. And the results will be analyzed by flow cytometry. Chronogenic potential will be measured using anchorage dependent and independent colony formation assays. The next thing I will look at is citrate oxidation, and this is because in normal mammalian cells, the glucose is converted, the pyruvate is converted to acetyl coenzyme A, which then reacts with oxaloacetic acid in the Krebs cycle to produce citrate, and this citrate is further oxidized in the Krebs cycle to produce energy. However, in the normal prostate cells are energy inefficient where the citrate is accumulated rather than oxidized in the Krebs cycle, and the citrate forms a part of the seminal fluid secretion. So, upon transitioning to cancerous cells, the normal prostate cells assume an energy efficient system where now the citrate is oxidized. And since acetyl CoA, which is the precursor for citrate, comes from glucose, I hypothesize that SGLT2 inhibition will lead to increased citrate levels. Citrate will be measured using the citrate assay kit, which is a luminescence assay that utilizes conversion of citrate to pyruvate by oxaloacetate, and this pyruvate is oxidized in multiple steps to produce luminescence. I will also monitor lipogenesis in PC3 cells. This is because in the normal mammalian cells, they fulfill their lipid requirements by utilizing free fatty acids from the bloodstream that is generated by liver and adipose tissue. However, cancerous tissues are able to generate their own lipids so that they can meet the increasing demand for biological membranes for rapidly proliferating cells. Additionally, PC3 cells exhibit high basal lipid synthesis compared to the normal prostate cells, PNT2. And, and since the precursor of lipid synthesis is acetyl coenzyme A, which comes from glucose, and since SGLT2 is a glucose transporter, I hypothesize that SGLT2 inhibition will decrease the lipid synthesis in PC3 cells. Lipogenesis will be measured by evaluating the C14 incorporation into lipids from C14 labeled glucose, and this will be normalized to DNA concentrations by ERGST assay of non-radioactive control plates. The potential problem in AIM-1 could be that different glucose transporters that are expressed or thought to be expressed in prostate cancer cells might compensate for the role of SGLT2 upon its inhibition. Some of the known glucose transporters that are expressed are GLUT1, GLUT12, and SGLT1. The expression levels of the GLUTs and other SGLTs upon SGLT2 inhibition will be measured by qPCR, and and upon figuring this out, the inhibition or extracellular knockdown of GLUTs or other SGLTs in PC3 cells will be performed to address this. So my aim 2 is to investigate the potential of SGLT2 inhibitors as a therapeutic for prostate cancer. The compartmentalization and heterogeneity in tumor microenvironment shapes the fate of tumor mass, where some cells in the tumor are highly glycolytic, whereas others are not. Prostate cancer death, additionally, occurs due to secondary metastases rather than the primary tumor itself. Thus, it is very important to effectively model prostate cancer progression in vivo in relevant animal models. Additionally, the in vitro findings, irregardless of their results, should be corroborated or contrasted in relevant animal models in vivo. As mentioned before, dapagliflozin, the SGLT2 inhibitor, has been shown to reduce tumor growth rate and increase tumor necrosis in pancreatic xenograft mouse model. However, whether SGLT2 inhibitors can show similar or play similar role in prostate cancer xenograft mouse model is not known and will be addressed in this study. Additionally, canagliflozin has been shown to reduce glucose uptake, clonogenic survival, and cell proliferation in PC3 cells. However, whether these in vitro effects can be translated in vivo are not known and will also be evaluated. To address this, orthotropic prostate cancer xenograft mouse model will be generated in Balbc athymic nude male mice aged 6 to 8 weeks. And luciferase expressing PC3 cells will be implanted into the ventral lobe of prostate. And tumor growth will be monitored using luciferase based in vivo imaging system. The therapeutic trial will consist of 5 different groups. The first is the placebo, the 3 different SGLT2 inhibitors, and a positive control, the anti-cancer chemotherapy drug gemcitabine. And around 10 mice for each group will be used. So before implantation of PC3 cells, the mice will be allowed to acclimatize in the treatment environment for around a week, so they are not stressed. And the PC3 luciferase cells will be implanted into the ventral lobe of prostate at week 0. And when there is around a 3 times increase in bioluminescence, suggesting tumor formation, the treatment will begin, which is estimated to be around 4 weeks. And the treatment will continue for another 6 weeks, upon which the mice will be euthanized for necropsy. A study done before generating this orthotropic xenograft mouse model has shown that lung metastases occur in 100% of the mice at around week 6 after PC3 cell implantation. So the treatment involves daily administration of 30 mg per kg of vehicle and treatment by oral gavage, and intraperitoneal injection of 80 mg per kg of gemcitabine will be performed once a week. Weekly submandibular blood collection will be done to evaluate the circulating tumor cells by using luciferase-based imaging and quantifying the PC3 luciferase cells. Tumor growth and metastases in live mice will be estimated weekly using the in vivo imaging system. Upon necropsy, tumor size and weight measurements will be taken, and hematoxylin and eosin staining will be done to analyze the tissue morphology. Cell proliferation will be evaluated by Ki67 staining of tumor sections, as mentioned before. And tumor necrosis and apoptosis will be evaluated by H&E staining, which can help recognize the early stage fragmented and small nucleus and late stage ghost cells without nucleus. I will also utilize the tunnel assay that utilizes the terminal deoxynucleotide transferase to add DUTPs to the double-strand breaks generated upon apoptosis and necrosis, and this DUTP will be labeled and will allow us to recognize the apoptotic and necrotic cells. Tumor metastases will be examined in the lung, bone marrow, lymph node, and blood. And the PC3 luciferase expressing cells will be identified using the luciferase based imaging. Similar to AIM-1, the potential problems could be compensation by other glucose transporters such as GLUT1, GLUT12, or SGLT1. To address this, I will evaluate the PC3 xenograft tumors for expression of various glucose transporters and SGLTs upon SGLT2 inhibition in vivo. And I will also do a knockdown of PC3 cells for glucose transporters or SGLTs to generate prostate cancer xenograft models. To summarize, SGLT2 is functionally expressed in human prostate adenocarcinoma, and the SGLT2 specific inhibitor dapagliflozin caused reduced glucose uptake in human prostate adenocarcinoma tissue as well as prostate tumors in PC3 xenograft model. Canagliflozin caused reduced cell proliferation, clonogenic potential, and increased cell death in PC3 cells. In pancreatic cancer xenograft model, SGLT2 contributes to glucose uptake, tumor growth, and survival. And this study aims to investigate the role of SGLT2 in altered metabolism of prostate cancer cells, as well as evaluate whether the FDA-approved SGLT2 inhibitor drugs could provide beneficial effects in PC3-induced xenograft models of prostate cancer. If this study will show that the SGLT2 inhibitors can provide beneficial effects in the prostate cancer xenograft mouse models, the future directions would involve figuring out the mechanism of action of drug and to evaluate whether SGLT2 inhibitor drugs used with other anti-cancer drugs can lead to synergistic effects. With this, I would like to thank my PI, my lab members, Derek and Simon, and my dear committee members.